So to start our cell chapter, before we actually talk about cells, we need to talk a little bit about microscopes because the knowledge that we have of cells really comes from microscopes because they're too small for us to see with the naked eye. So there are three factors involved in the quality of microscopes. The first one is magnification. In other words, how much can this microscope magnify the image? 10x, 30x, 100x, 1000x. How many times bigger can we make the image? The second is resolution. Resolution is a little more difficult to understand, but resolution is the distance between 2 points that you can still distinguish them as two separate points. For example, if I draw these two little dots on the board, if you were to walk back far enough from your computer, these two dots would probably blur into one. You wouldn't be able to distinguish that they were two dots anymore. And that's resolution, the ability to see between two things that are really close together. And honestly, this is probably the most important and the most limiting factor of microscopes. If you go to a hobby store, you can find microscopes for 50 bucks. And they're going to tell you that those microscopes magnify 400 times. The truth of the matter is, yes, they have the magnification, but because of the way they're made, they don't have the resolution. So when you go to try to look at something at 400 times, what you're going to find is it's going to be very, very difficult to focus, and you're actually not going to be able to see the details. Even though it's magnified, the details are still going to be blurred. And finally, contrast. Contrast is the ability to see different parts of the sample sort of standing out. And I have actually a really good slide here of contrast. So this is the same exact picture, but they used a technique called fluorescence, where they basically tagged the cell with radioactive markers. And if you recall from our last couple of chapters, when we talked about what a radioactive isotope was, radioactive isotopes give off, because the nucleus is unstable, they give off radiation, and that can actually be seen in the form of a glow. So by radioactively tagging these cells, we've actually improved the contrast and the resolution. We can see between the parts better, and the parts really stand out. And so the same, we haven't changed the magnification here, but we've been able to improve especially the contrast. Okay, so this is sort of a quick summary of the three types of microscopes that we talk about in here, and the three types that are really used to get pictures of cells. So the first one is the light microscope. This is what we'll be using in class. Now, there's a lot of information here, but for testing purposes, I'm going to highlight what you actually have to know. So the light microscope uses visible light. Notice both electron microscopes use electrons. In other words, in the light microscope, we actually look through a lens, light shines in from a lamp, and we actually look directly at the image. We can use it to view live organisms, which you cannot do with the electron microscopes because they have to be stained with metals, and there's a whole preparation process that goes on. And we can only magnify up to about 2,000 times. And technically the ones we have in class, we're only going to be able to magnify up to 400 times. But even the best light microscopes only magnify up to 2,000 times. And you do need to know the reason why. And that is this, down here. You see, remember that light, there's different wavelengths of light. And you may remember from chemistry, visible light was then the range of red, orange, yellow, green, blue, violet. And the smallest wavelength of light that our eyes can actually detect is about 400 nanometers. Any light that has a wavelength shorter than that, our eyes can't see it. So if we're relying on visible light to basically light up our image or illuminate our image, what's going to happen is any details that are closer together than 400 nanometers, we're not going to be able to see them. They're basically going to blur. And so what we have is a limit on the resolution. In other words, we could make lenses that magnified more than 2,000 times, but the bottom line is everything would just stay blurry. We wouldn't be able to focus it because of the resolution issue. Now, electrons, on the other hand, travel at, they have wavelengths much, much, much smaller than visible light. And that means by having those tiny, tiny wavelengths, they can capture details that the light microscope cannot capture. So, what do you need to know about the light microscope? It uses light. We can see live organisms. Its maximum magnification is about 2,000 times, and the reason why we can't magnify more than that is it's a resolution issue. It's because of the wavelength of the smallest wavelength of visible light that we can see it's about 400 nanometers, and when you get smaller than that, any details that are smaller than that, the light's just going to blur. We're not going to be able to see them. Okay, now for the 2-electron microscopes, you do not have to know how much they magnify exactly. What I would like for you to know is that the scanning electron microscope is the one that gives you 3D images of the surface. And if you look at the picture, you can see this is the same paramecium under all 3 microscopes. Notice how under the scanning electron microscope, you can see all the little hairs on its surface. And then finally, the transmission electron microscope, it actually is the one that lets us see all the cell parts. This is the microscope that really allowed us to identify the mitochondria and what's inside a mitochondria and what do ribosomes really look like. What's inside of the nucleus, because this one magnifies up to 10 million times, and you don't have to know that, but you should know that this is the one that magnifies the most. That if you want the microscope that's going to magnify cells the most, the transmission electron microscope is going to be the one that you would want to use. Now, we don't have them here. For one thing, they cost like over $100,000. For another thing, they're humongous. They take up like a whole corner of a room. And you would have to probably take a 6-week course just to learn how to use the microscope and how to prepare the slide. So it's not sort of a plug-and-play kind of thing. They're very complex. But the transmission electron microscope in particular is the one that really allowed us to learn what the ultrastructure of cells was. What was really inside of a cell. Because we could really see the details. So those are our 3 kinds of microscopes. Here's a picture that compares the scanning electron to the transmission electron, and you can actually see the difference there, how the scanning scans the surface and gives you that 3D picture. And then the transmission electron microscope actually allows us to see inside the cilia, which are little hairs. You actually have cilia lining, for example, your respiratory tract. Okay, the next section is on cell size, and I believe my other recording should overlap this. Cells are usually 2 to 200 micrometers in size. We're going to talk about metrics in another lecture in a minute. Viruses are measured in picometers or nanometers, which are even smaller. Too small for us to see with the naked eye. And as you can see from the diagram, light microscopes, when they were first developed, allowed us to see plant and animal cells and bacteria a little bit. But in order for us to actually see really tiny things like viruses and the tiny structures inside of cells, that required the development of the electron microscope. All right, and then what limits cell size is the surface area to volume ratio. As a cell gets bigger, both its surface area and the volume, they both increase. But what happens is the surface area does not increase as quickly as the volume does. So as a cell gets bigger and bigger, what we end up with is a smaller surface area to volume ratio. And in order for a cell to be effective, it needs a big surface area to volume ratio. So the bigger the surface area to volume ratio is, the more efficient a cell is going to be. And the bottom line is that, remember what surface area and volume actually are. Surface area is the exposed surface. In other words, anything that has to get into the cell has to do so through that surface and get to the middle. So the more surface area I have, the better that cell is going to be. On the other hand, if I have a very gigantic cell, I do have more surface area, but I have more volume too. And in fact, the ratio is smaller. I basically have less surface area compared to volume. And the bottom line is stuff just can't get in and out fast enough to keep the cell alive. And so that's why we're made of millions of little tiny cells rather than just a few really large cells. So in the next section that I'm not going to go into now, but you can check the video on surface area to volume ratios. I'm going to give you a walkthrough of how to calculate surface area, volume, and the ratio. I'll leave you with this slide here just showing that if they were to give you 3 or 4 cells that were all cubes, or 3 or 4 cells that were all spheres, and they said which one has the best surface area to volume ratio, you would not have to do any math. You could answer this question immediately. Because since the rule is that volume increases faster than surface area to volume ratio is going to go down as the cell gets bigger. So this sphere is going to have the best ratio. This cube is going to have the best ratio. But if they give you, say, a cube and a sphere and they want you to compare them, that's where you would actually have to do the math and do the calculations. And that's what is in the video called surface area to volume ratios.